Evet, 9 Eylül Üniversitesi'nde çalışıyorum.
I'm from 9 Eylül University. I'm also a part of the Bipolar Disorder Consortium of Harvard University, and I am the head of the Mood Disorder Association. As explained by our moderator, I will try to give you some information about PKC and GSK3B pathways as potential new treatment targets. This is quite a complicated slide to just show us what we have been inspired by. I had prepared this slide years ago. It is a good recap of what we know. Lithium and valproate and their effects on signal transduction systems can be shown on this slide. Of course, we don't know all the mechanisms behind their effects, but there are ongoing studies for the last 20 or 30 years about the possible mode of action of lithium and valproate. This is basically what we know so far. And we have taken this path to cover some distance and to learn more. Lithium and valproate are effective or interact through protein G, the phospholipase system. Protein kinase C is activated at the end of this pathway by both drugs. Glycogen synthetase kinase is activated especially by lithium. There was some data pointing to the fact that valproate inhibits it, but that data was never replicated. Lithium inhibits GSK3 beta effect. There are some other factors. There's also PKC substrate, and that is also another information that we have about these pathways. What about our targets? Let me give you some other targets. We have PKC inhibitors, GSK3 beta inhibitors, and we have oxidative damage removers. This brought about a significant change in the understanding of bipolar disorder. Pronergic system and tachykinins have become topical. HDAC inhibitors and melatonergic systems are being studied as well, and glutamatergic system is also being studied very frequently. Let me go through them one by one. Oxidative damage removers are very effective because they have an important role in the circuits. If there's a problem in the energy system, this can be fixed by these agents. It was believed that in the bipolar disorder, there might be a problem with the energy system. Therefore, certain agents such as cysteine glutathione precursors and regulators are being tested. And acetylcysteine glutathione increases glutathione. There are certain bits of information. Glutamate's cysteine ligase modifier is another agent that is being studied within the framework of this mechanism. We also said that the pronergic system and tachykinins have been studied recently. ATP, adenosine dopamine and adenosine regulate dopamine, serotonin, and GABA activity. ATP-P2 antagonists might have antimanic effects. They're being studied for that. Adenosine 1 and 2A agonists are being studied for their sedative, antipsychotic, and anti-aggressive effects. Allopurinol has a pronergic modulator effect. And of course, there are tachykinins, as I said before, substance P, neurokinin A and B, and the receptors that they have an impact on. On receptors 1 and 3, there is not much of an effect actually, but neurokinin 2 might have antagonistic effects. There are some studies pointing to that direction. HDAC inhibitors and melatonergic system are also mechanisms that are being studied. Valproate inhibits acetylase at the histons. Histons are, of course, very long. I don't remember the exact length of the DNA, but there's a perfect packaging system which makes it fit into the nuclei. And histons, of course, have a very important role to play in this packaging process. The acetylation of the histons keep them from binding to the DNA, which regulates the gene expression. And it is recently believed that this has an impact on the epigenetic mechanisms. Therefore, histones are very important for the epigenetic mechanisms. Valproate inhibits the histone deacetylase. Maybe that is why it has an impact on the bipolar disorder. Melatonergic system is very topical as well, especially on the circadian, thanks to the data on the circadian cycle. Therefore, valdoxan is being studied, and agomelatin is another agent that is being studied within this framework. Before I move on to PKC, I would like to talk about cellular plasticity augmenters or enhancers and glutamatergic system. On Saturday, we're going to hear a speech about the glutamatergic system from one of the professors. He will talk about the neuron-glia cycle having an impact on bipolar disorder. There are, of course, many scientists who discovered data on this. Of course, the protection boils down to some neuron-sparing mechanisms. B-cell lymphoma protein 2 is one of the most important factors in this, as well as BDNF, for example. AMPA enhancers are very important. They increase BDNF. NMDA antagonists are very important. Pramipexole is important in increasing BCL, in enhancing BCL. Riluzole increases BDNF, therefore it might have an antidepressant effect, but there are no clear-cut data that point to this direction. There is some negative data, actually. Ketamine is an NMDA antagonist. It has an antidepressant effect. The data has been replicated as well. However, the effect wears off after a while, but it's one of the mechanisms that is being studied. Glutamate is toxic, therefore there are also glutamate release or secretion inhibitors. Corticotrophin releasing factor and glucocorticoid antagonists are also studied as possible mechanisms. These are the signal transduction systems, CLAP, BLP, BCL2, protein kinase A, and MAP kinase system are some of the neurotransmitters that we're used to hearing about. Now I will move on to our main topic, GSK3 beta. GSK3 beta is a very important protein. Valproate, for example, is found to have no impact on GSK3 beta. GSK3 beta regulates PI3 kinase WNT, PKA, and PKC activities. So these are mini proteins that have an impact on transduction within the cell. And thanks to this regulation, it has an impact on glycogen synthesis, gene transcription, synaptic plasticity, apoptosis, and some other mechanisms. GSK3 beta inhibitors have already been tested on animal models and have been shown to have an antidepressant and anti-manic effect. However, so far we have not discovered a GSK3 beta inhibitor that passes the brain-blood barrier. Carbamazepine and valproate do not seem to have an impact on this system. GSK3 beta has an impact on beta-amyloid plaques that are formed in Alzheimer's. Therefore, this is one of the mechanisms that is being studied to find a cure or to find a treatment to Alzheimer's as well. We have lots of data about PKC. This phosphatizes the serotonin portion of many proteins. It has 8 homolog isozymes: alpha, beta, beta 1, beta 2, gamma, delta, epsilon, C, and E. Normally within the cell, it is inactive in the cytoplasm of the membrane, then it moves to the membrane when it's activated. It is activated through diacylglycerol pathway. Lithium and valproates inhibit the PKC activity. It has an impact on neuronal accessibility, anti-release, ion channels and receptor activity, gene transcription, and gene translation. Therefore, it also regulates cell proliferation, apoptosis, and migration, for example, as well as dendritic spine morphology and plasticity. Therefore, protein kinase A became a very interesting pathway to study for many different diseases: cardiac failure, stroke, diabetes, pain control, are some of the diseases that is being studied for. PKC has been tried in many different trials. However, most of the trials were concluded at around phase 2 because the data for other diseases other than bipolar disorder are not very conclusive. Probably it has to do with its non-selectivity. But in bipolar disorder, this is not true. This is one of those areas where very concrete data about PKC comes together. I will try to briefly summarize the data. Well, the oldest data was found in the platelets of the bipolar manic patients. In the platelets, PKC activity was detected to be high, and this was reversed either by valproate, for example. And this was proven in studies as well. This is the control group, cytosol and membrane-bound activity. It is high. This is the control group. This is the bipolar group. A 2-week valproate study shows us that the levels are reduced. The control levels are reduced. It was also detected that PKC increased as a result of serotonin secretion. I won't go through all the items one by one, but this has been replicated many times. I had talked about the Marx protein before. The Marx protein is also regulated through lithium and valproate. So therefore, the researchers say that this also points to PKC. Psychostimulants that lead to manic attacks in humans and in animals activate PKC, and valproate reverses this effect. An important animal study was carried out by Aynat and colleagues and at NIH. Tamoxifen is a PKC inhibitor, and it has antihistamine effects as well. In an animal model induced by amphetamine, the risk-taking behavior and hedonic behavior were normalized. This is another important study from Science that was published in 2006 in apes and mice. Exposure to stress increased PKC activity. Overexpression of the PKC impaired cognitive function at the prefrontal cortex at both animals. Another study from NIMH with promanic amphetamine therapy, PKC activity was doubled in the frontal cortex of mice. Psychostimulant amphetamine therapy increased the phosphomarx levels by twofold as well at the prefrontal cortex. Marx phosphotation also points to PKC activation. This is some additional data from the same study. In the mania model, as a result of sleep deprivation at the prefrontal cortex, PMARX levels were increased by 1.5 times, and antidepressant imipramine therapy also increased the PMARX levels at the prefrontal cortex. With 4 weeks of lithium therapy, this was reversed. Let me go on with the same study. Neurogranin is another PKC substrate, just like Marx. Neurogranin is found to be a PKC precursor. Therefore, its phosphorylation increased significantly through promanic manipulations at each of the 3 promanic manipulations. Lithium therapy normalized the levels of neurogranin as well. This is from another study in mice that received imipramine and who were sleep deprived. PKC activation increased at the NMDA receptors, and lithium therapy reversed this case. Another important finding from 2 different genome-wide association studies. DAG kinase eta was found to be a risk gene at bipolar disorder. DGKH is the main regulator of DAG, which is the main regulator of PKC. PKC inhibitors tamoxifen and chlorythrin can go through the blood-brain barrier. When we test tamoxifen, usually we administer clarifen as well to see where the effect comes from. Hyperactivity and risk-taking behavior were reduced significantly. There are many more studies that bring about consistent data from animal studies. Tamoxifen was found to fix the impaired PKC activity in animal models. One final study in 4 different parts of the brain. Manic behavior was induced through amphetamine, and it was fixed with lithium and tamoxifen therapy. PKC activity was reduced as well. Phosphocrep and NGF and BDNF levels were checked as well. And it was found that with amphetamine therapy, these, as you can see on the slide, were reduced. The BDNF levels were reduced. With lithium and tamoxifen, this impairment was fixed. If lithium and tamoxifen are given as early as possible, then the impairment can be prevented. Otherwise, it can improve the impairments. There are 6 clinical trials in total. 4 of them are add-on therapies. They're all placebo-controlled, double-blind, randomized trials. Only one of them is not a double-blind, but a single-blind trial. I will first talk about the 2 add-on studies. One is from NIMH on 7 patients, and the second one is from Australia on 13 patients. They both brought about positive results. The antimanic effect of tamoxifen were tested in a clinical setting, and they all gave positive results. This is investigator-initiated group of concept study, a phase 3 study. 2 different studies again, one from NIMH. These were started actually simultaneously. One was published a bit earlier than the other. Zarat and colleagues published the first article on this. They worked on 16 patients, and in Turkey we conducted our study on 66 patients, and we were funded by SMRI. The baseline mania scores were approximately 25 in the Zarat group and more than 30 in our group. The rate of psychosis was 50% at Zarat and 66% for our group. These are quite high rates for investigator-initiated study, for studies of this kind. At investigator-initiated studies, you don't see such high rates, for example. The rate of mixed episodes might also be high, but it has to be kept under control. At the Zarat study, it was around 33%. In our study, the rate of mixed episode was only 6%. But we will see the findings of both studies very shortly. And they're both very strong data, very robust data. Archives of General Psychiatry was the journal where we published our patients. The weekly improvements was in the mania score was significant. It fell from 38% to 20%. Same goes for the tamoxifen group. There was a decrease in the tamoxifen group, but placebo remained the same. There was an improvement both in the young mania scores and the PANSS total scores of the tamoxifen group. It was significant, but placebo group remained the same. Lorazepam was given as a rescue medication because we're talking about a group where there's a very high rate of mania. This was, of course, limited in the couple of first days of the protocol, but then lorazepam was administered as well. This shows the weekly exposed dose of lorazepam. Intent to treat results. Well, the difference between the 2 groups is very significant and is pronounced as of week 2. Same goes for PANSS. Here you see the response rates, 48% to 5%. So there's more than 50% of improvement at the young mania scale. We haven't run into any significant or severe adverse events in both groups, the tamoxifen arm and the placebo arm. There was a suicide attempt by one patient. Acne reactions and redness on the face were some other findings. NIMH study studied 16 patients. And the improvement of the tamoxifen arms in both studies was very consistent. We were independent studies, though, so we didn't know about their data before it was published. mean change in the Young scale was identical, 18.3%, and the deterioration at the placebo group was slightly different between the two studies. And here you see the response and remission rates for both studies. This is also quite a consistent finding. So as a result, we can say that there are no clinical trials on GSK3B. However, there are preclinical studies that give us an idea about the antidepressant and antimanic effects potentially. Both preclinical and clinical data are very robust and consistent. However, we have to replicate the data with higher numbers of patients. Tamoxifen therapy is considered to be a third-line therapy in the JANMAT guidelines, and I agree with the guidelines, but in the long run, if we were to look at the benefit-risk ratio, then tamoxifen should not be considered because it might increase the uterine cancer risk and the risk of thrombosis as well. But it passes the blood-brain barrier, and we have to develop some new molecules that are effective on PKC alpha and PKC